Hello bio students, this is Mr. Yago and I'm going to dive into section 12.3 meiosis. Now meiosis is a little bit different than mitosis. A lot of the same rules apply, so it's a kind of a great review of mitosis, but it's definitely going to go into a lot more detail regarding each of these steps, and it happens twice. So pay attention, take good notes, and we'll get after it. Alright, so how does meiosis differ from the processes of mitosis? What you're going to notice right off the bat is that we actually go through the process of mitosis twice. So there's actually what we classify as a meiosis 1, and then we have a meiosis 2. And some interesting facts is it does not duplicate the chromosomes after the first division. So if we go through meiosis 1, that first section, we do not duplicate chromosomes. And our real goal here is that we are trying to have the chromosome number from 2n to n. And I'll explain what that means in more detail later. Now, the reason that we have to go through this process is because we're looking at mixing genetic material from both the maternal or female side and the paternal or father side. And what that does is it's going to give us genetic variation. That's why you look a little bit like mama and a little bit like dada. And if you look up here, this is the process that we're going to talk about, something called crossing over. And if you look at this picture, before and then after, you'll notice that there are some definite changes. Now, this crossing over is where we exchange corresponding pieces, meaning that, let's take a look at this top section here. That could get exchanged. This could be mama, this could be dada, and then you have the ability to go ahead and exchange and thus give you some of that genetic variation. So the traits that you have today, like if you have your mom's eyes or your dad's hair color, that all has to do with the fact that when your chromosomes, when your parents' chromosomes, specifically the sex chromosomes, the sperm and the egg, are developed through the processes of spermatogenesis and the process of oogenesis, the making of sperm and egg, that we went through this process to give that variation upon conception. All right, so the first thing right off the bat, and I apologize for the picture, but this step is prophase 1. What's unique about this particular step is the fact that we still go through chromosomes condensing, we still have nuclear membranes breaking down, but that homologous chromosomes pair up and go through this process of crossing over. So on the previous slide, I mentioned how important crossing over is for what we classify as genetic variety. So when we go through this mixing of genetic information, it's happening in this prophase 1 as we classify it. Because remember I said that in meiosis we have 2 steps. We have meiosis 1 and meiosis 2. So the normal things occur, chromosomes condense, membrane breaks down, but this crossing over is imperative to understand how we get that genetic variety. All right, our next step is metaphase 1 in this meiosis 1 process. And there's a couple things that I want to bring to your attention. One is the fact that, as usual, we're lining up on the metaphase plate, and our spindle fibers are responsible for attaching to those centromeres, which you can see right there in white. But there's an important part that I do not have written down, but I am going to quick write down as well, but that's the fact that homologous chromosomes line up. So these are considered homologous chromosomes, and they're going to go through a process called independent assortment. Okay, so I'm going to write that one down, independent assortment. And basically what that means is that they will randomly line up. Okay, so they're going to randomly line up on that line, and what that's going to do is it's going to essentially allow for some genetic variation. Okay, so you'll notice that the colors are a little bit different. We have a blue and a white and a blue and a white. So these actually swapped positions, and they will go to that side of the cell and that side of the cell and eventually begin in anaphase and telophase to separate into those cells. So independent assortment is a big thing to know, which means they basically line up randomly on that metaphase plate and allow for a little bit more genetic variation. Anaphase is the same thing. Now that we have our homologous pairs lined up, you'll notice that the spindle fibers that have attached are now ripping those apart and pulling them to what we classify as the poles of each side of this cell. I also want you to notice the shape is changing, because remember, the end result is we want to try and make two cells out of this. So as it starts to, expand in these directions, it will then go through the process of cytokinesis. So here's telophase 1. Again, notice that we have the chromosomes gathering at the poles. One thing that I don't mention here is that you'll notice the nuclear membrane is starting to reform. It was broken down, but it's starting to reform around those daughter cells that we made. But the one thing to notice is that these are haploid. These are haploid, so they actually have half of the genetic information. So this was full, total, and now we split it in half. So now these are considered haploid cells. All right. Also notice that this is cytokinesis, so the cytoplasm will divide. So basically the goal of meiosis 1 is to cross over, get that genetic variety, go through the process of metaphase, anaphase, and then in telophase here, we are going to break this apart into two cells and basically doubling. All right, so now we've gone through meiosis 1. Now we're going to dive into meiosis 2, and again, we start with the same thing. We start with that PMAT, that prophase, metaphase, anaphase, telophase. Now what's unique about this is that we have two haploid cells, which means that we have half of the DNA. 23 chromosomes set, or 23 chromosomes, 23 chromosomes, and we have that haploid set. A total full-bodied diploid would have 46 chromosomes. But a new spindle fiber forms around those chromosomes, and centromeres move towards the pole. So they don't show it in here, but within this cell, I got it marked right here, you'd have your centromeres here, your centromeres here, and then you have your attachment site of that spindle fiber. So again, we're setting up the stage. Now metaphase, same thing. Notice that we have them lined up. Here's your centromeres. It shows it a lot better than on the previous picture. Your spindle fibers are attached, but we're lined up on that equator, and as you can predict in anaphase, we're going to start pulling them apart. So here we are in anaphase 2. The centromeres divide, and notice that we have now actually split into chromatids, which is half of that chromosome. So we are actually taking that DNA information and cutting in half again. So again, we have two cells here. The goal of meiosis is to essentially make 4 haploid cells. All right? And I'm going to explain why that haploid is important. So the centromeres divide, the chromatids separate, and don't get that confused. That's a chromatid. If they formed an X, that's a chromosome. So these are chromatids, and they're going to move to the opposite ends of the pole. All right, in our final telophase 2, you'll notice that the nuclear envelope forms again around each set of chromosomes. And really that should really say chromatids. Chromatids. So make a note of that, chromatids. And then the cytoplasm will divide. Now, what we call daughter cells, we actually have 4 daughter cells that are going to be generated here. So if we're a male and we're going to make sperm cells, we start out with 1, breaks up into 2 in meiosis 1, and then it actually makes 4. So every time we go through this process called spermatogenesis, we're making 4 sperm cells. So I'm kind of bummed out because this was supposed to animate and go through the whole process, but I'm just going to kind of wrap it up and speak to the fact that our goal for meiosis is just to make sex cells. So we're just making sperm, we're just making egg. That's the goal. The only difference is we're going to find out is that the amount of each that we make is a little bit different. So remember your PMAT, remember that order, remember the major concepts. The major things that are different are going to be found in that very first slide of information you took, and that's, I think, is kind of the most important part of this whole process, because meiosis is the same thing as mitosis. All right, last but not least, these are kind of these big differences, and I'm just going to speak to this real quick. So in most male animals, we generate 4 equal-sized sperm cells. We started with 1. Notice the chromosome numbers here. So we actually cut it in half. So this is diploid, 2 haploids, and actually we doubled those haploids to make 4 haploids. So there's 2 divisions. This would be meiosis 1. This would be meiosis 2. Now, here's the cool thing. So when guys make sperm cells, they have a tendency to, and usually it's 50-50, but there is variation. Some of these will be X, Some of these will be Y. Okay? Now, a male, if we're looking at the sex gametes, the 23rd chromosome, if it's XY, that means it's a male. If it's two X's, that'll be a female. Now, females only make X. Okay? So if you have an X and an X come together, guess what? You're having a girl. If you have a Y and an X come together, if the Y sperm is the one that makes it in and fertilizes, well, guess what? You got a baby boy coming. So it's kind of cool. So basically, the father is going to determine the sex of your baby. And I know it sounds kind of weird, but it's a 50-50 shot. Either you get an X sperm or a Y sperm, whatever one makes it in and fertilizes the ovum, is going to determine what kind of baby you have. Now, in females, most of the cytoplasm remains as one, and we call that an ovum. So just like we call this spermatogenesis, oogenesis, or oogenesis, which is oogenesis, you end up with one really large cell, which is going to be the ovum, and then three what we call polar bodies. They are not functional, and eventually they're going to break down and disintegrate later, but actually the ovum will kind of use these for energy. So it's almost like if you had quadruplets, if you were a mama and you had quadruplets, and then one baby says, "Oh, I'm so hungry, I'm going to eat the other babies, nom 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 nom." That's kind of what happens. Cool. Well, if you got questions, give me a shout, and these are the steps of mitosis and what I feel is the most important things. So, Yago, out.